afternoon. Okay, it is our great honor to have Dr. Angelika Amon come to talk to us today. Who is Dr. Amon? She got her PhD from the University of Vienna, where she worked in Dr. Kim Naismith's lab. After she became a Whitehead Fellow, and following that, Professor of Biology at MIT. And now she currently is a Howard Hughes Medical Institute investigator. She's been awarded many prizes. The Alan T. Waterman Award, which was an act of Congress to be awarded to young scientists who are under the age 35 or no more than 7 years out of their PhD. The E.I. Lilly Young Investigator Award, the Paul Marks Prize for Cancer Research Award, the National Academy of Science Award in Molecular Biology. And this is a celebratory event because just last week, Dr. Amon was inducted into the National Academy of Sciences. At MIT, Dr. Amon's lab focuses on two major themes: the regulatory networks that control chromosome segregation during mitosis and meiosis. And she also is studying the effects of aneuploidy on tumorigenesis. So enough of me.
Thank you very much. It's a great pleasure to be here. My first time at Davis. It was a truly splendid experience. I had great interactions meeting with the faculty. I met 3 or 4 one-day-old cows during my visit also. That was also quite special. You have a wonderful campus here. Congratulations. It's very nice to be here for a visit. And so as Sean said, my lab's been interested for many years in trying to understand the molecular mechanisms that ensure that chromosomes are segregated accurately, or in other words, the molecular mechanisms that ensure. Do you want me to close the door maybe? Yeah? Okay. Let's get a little more cozy now. Trying to understand the molecular mechanisms that ensure that aneuploidy, a state of incorrect chromosome number, doesn't happen. But several years ago, we decided to sort of go a step forward and ask, well, what actually happens to a cell where these safety mechanisms, these surveillance mechanisms fail, and that actually do end up with the wrong chromosome number. And these studies are the ones that I'm going to tell you about today. I'm sort of going to tell you about our efforts to understand, well, what really are the consequences of aneuploidy. And before I do this, I want to sort of get some definitions straight, because a lot of times in the literature there's some confusion. So basically, what we define, aneuploidy is defined as a chromosome composition that's not a multiple of the normal composition. And I want to make a point here that this is actually very distinct from polyploidy, where the entire genome has been duplicated. And what I'll show you, what I hope I'll be able to convince you of by the end of this talk, is that the problems associated with aneuploidy are actually quite distinct from those that are associated with polyploidy. In a nutshell, aneuploidy, we believe, is all about problems in protein stoichiometry imbalances. And obviously, these problems do not occur in polyploid cells. And so the study of aneuploidy is sort of one that has a very long tradition. The first time this has been studied systematically was in the developing sea urchin embryo by Theodore Boveri. And sort of he asked, well, what are the consequences of aneuploidy on the development of this sea urchin embryo? And in a nutshell, what he found is that aneuploidy is bad news. It causes developmental defects, and in most cases cell death. And this sort of picture depicts of what he saw in the microscope. Basically, what he did is he took a sea urchin egg, and fertilized it with 2 sperms. And that obviously then will lead to the formation of what's called a tripolar instead of a bipolar mitosis, and massive aneuploidy ensues during that first chromosome segregation. And he then basically cataloged what he saw. So this is a normal or almost normal gastrulating sea urchin embryo. And these are the messes that he saw, or better actually his wife saw under the microscope. As in so many cases, a lot of the work is done by the better half of the pair. what she saw was like these severe effects on aneuploidy. And I should say that we know today that in every organism where this has been studied, aneuploidy has severe consequences. And here's another one of these examples, another very famous example by Barbara McClintock, where she created these maize plants that have one additional chromosome. And as you can see, what she observed is that in these maize plants, these carried an extra chromosome, they were growth retarded and runted. And so, of course, also in humans, we know that aneuploidy has severe consequences on fitness. Of course, we know that all autosomal monosomies and most autosomal trisomies in the humans are lethal. Aneuploidy is the leading cause of mental retardation in humans, and it's also the leading cause of miscarriages in humans, with almost a third of miscarriages being due to aneuploidy in humans. So very clearly, at the organismal level, aneuploidy is bad news. However, we also know that at the cellular level, the situation may or may not be different. But a fact is that aneuploidy is obviously also associated with a disease that is characterized by uncontrolled proliferation, that is cancer. So in humans, for example, more than 90% of all solid human tumors are actually highly aneuploid. And sort of these two observations obviously produce a very interesting conundrum, right? On the one hand, a presence of a single extra chromosome is highly detrimental, in most cases lethal. Yet at the cellular level, very frequently we see severe karyotypic abnormalities associated with a condition where cells proliferate with great potential, much better a lot of times than normal euploid cells. So you can make an argument, and there's certainly a value to this argument. You can argue, well, making a brain and making a heart is complicated, right? And if there are some key developmental factors, and if there's imbalances in the stoichiometry of some key developmental factors, you get malformation of an organ, the brain or the heart, and that obviously leads to lethality of the organism. And at the cellular level, it doesn't really matter how many chromosomes you have, as long as you have one of each. So you can make all the essential proteins, and maybe more actually at the cellular level is better. Maybe then they grow better. The alternative hypothesis is that perhaps aneuploidy is actually bad news for the cell too. But in some way that we don't really understand is that these cancer cells have actually learned to live with the bad effects of aneuploidy. And so what we thought was going to be very important is what we wanted to do is sort of address this question is we needed to understand what the consequences of aneuploidy on normal cell physiology is. And that's actually several years back we set out to do, and this is what I'm sort of going to tell you about today. I'm going to tell you about our question, about our efforts to understand, well, what are the consequences of aneuploidy on cellular physiology? I'm going to tell you sort of efforts on two fronts. I'm going to initially tell you about our efforts of characterizing aneuploid yeast, budding yeast strains. We created 20 different defined aneuploidies in yeast and characterized these phenotypes. And I will also tell you about efforts to understand the effects of aneuploidy on primary mammalian cells. I'll tell you about our studies of 5 primary mouse embryonic fibroblast lines that are trisomic for one of 4 different chromosomes. However, before I do that, I would like to sort of address one question a little bit before I sort of tell you about the data. I want to sort of bring up one more point, and I think that is quite a valid point. And that is sort of, you should ask the question, well, is disomy or trisomy really a good model for aneuploidy in cancer? Right? Because normally, the karyotypic abnormalities that you see in solid human tumors are actually not just 1 or 2 extra chromosomes. They are always, most of the time, almost near tetraploid. So very severe aneuploidies. So obviously, we think that it is a good model, and I'll give you the reasons, but obviously, in the end of the day, you'll have to decide for yourself. So the trisomies and the disomies that we study, they're obviously very defined. They do not change, and all the cells are the same. So that's an advantage. You know what you're working with, okay? what I'm going to tell you is that actually many of these different aneuploid cells or these disomic cells and trisomic cells that we look at actually share a number of characteristics, and we're actually very excited about those, and I'll tell you in a minute what they are. And obviously what we postulate is that the characteristics that are shared by many different disomes and trisomes, they probably also exist in cells with severe aneuploidies. But as I said earlier, however, you will now argue cancer cells usually exhibit severe aneuploidies, and they're usually near tetraploid, and that led to the idea that most tumors are tetraploid first and then sort of start losing some chromosomes and end up at a near tetraploid aneuploid state. So that is definitely true, but sort of the late stage tumors that usually get resected, that is definitely true with the karyotypes. But I would like to remind you, or if you haven't known before, tell you that that's probably not how many tumors start with. So there's very good cytological evidence from pre-malignant lesions, and the best evidence that exists is for breast and colon, that show actually low level of aneuploidy, 1 or 2 extra chromosomes in these early pre-malignant lesions. And sort of based on these observations, we would argue that actually does have merit to look at these sort of disomes and trisomes and then hope to learn from them and try to extrapolate on tumorigenesis. So this is the thought process that was involved in sort of deciding to study these disomes and trisomes, and that's what I'm going to tell you about today. I'm going to first tell you about the characterizations of the effects of aneuploidy on normal cell physiology, and I'm going to show you that aneuploidy is deleterious at the cellular level, hands down, okay? And if the only thing you take away from this seminar is aneuploidy is bad news for cells. And I'm actually going to show you that aneuploid yeast and mouse cell actually share a number of traits that are indicative of proteotoxic and energy stress. Then obviously I'm going to sort of explore this idea further that perhaps aneuploid cancer cells have learned to deal with the stresses that are associated with aneuploidy. I'm going to tell you about our efforts. They're ongoing and it's still early on, about strategies to identify mutations that suppress the adverse effects of aneuploidy. I'm going to tell you that we've identified a deubiquitinating protein, UBP6 in yeast, that actually suppresses the adverse effects of multiple aneuploids. And in the last part of my talk, I'll sort of tell you that the flip side of this question is that can we exploit the aneuploid stage to find conditions that selectively kill aneuploid cells? And I'm going to tell you a story from our mouse effort. I'm going to tell you that we've identified an agonist of a protein kinase that's important in energy metabolism, ICAR, that appears to have these properties. So let me first start out with the first point. Let me tell you about our characterization of these aneuploid yeast strains. And I'm going to spare you the rather baroque genetics that is required to generate these aneuploid yeast strains. Basically, this is a CGH analysis where we quantify the genomic content of individual yeast strains to demonstrate that indeed these cells do carry an extra chromosome. Each of these boxes represents the karyotype of a particular yeast strain. And I hope you appreciated that there are stretches of the genome that are amplified twofold. And if I tell you now that this genome is organized by a genomic position, with the left arm of chromosome 1 on the left end of the box and the right arm of chromosome 16 at the right end of the box, I hope you appreciate that these stretches correspond to entire chromosomes. So we've created 13 of the 16 possible disomes in yeast, and then by accident, sometimes we get disomic strains that carry more than the chromosome that we selected for. And in some cases, we actually understand why that is the case. So even though we're able to select for both of these chromosomes, they are stable enough so that we can carry out a basic characterization of these. And as I said to you in the introduction, what we found is very clear. In yeast, aneuploidy causes a proliferative disadvantage. And to yeast geneticists, to sometimes, get a disome or a trisome into their crosses, you kind of already know that you probably are not very surprised by that finding. But we were, so we kind of expected that. But we were surprised to see that aneuploid cells actually did appear to share a set of phenotypes. And what I'm going to show you in the next few slides is that we could show that these phenotypes are actually due to the proteins that are being produced from these additional chromosomes. So this is a summary of the phenotypes that we found in these yeast strains. They have a proliferation defect, and we could show that the majority of them have a defect at the G1S phase transition. There's a transcriptional response associated with the aneuploid state. We have evidence to suggest that there's an increased need for energy in these aneuploid strains, and perhaps the most telling, or to us most striking set of phenotypes that we observed was that these aneuploid cells appear to experience proteotoxic stress. And this proteotoxic stress manifested itself by the fact that all 20 different yeast strains, or most of them, were sensitive to conditions that interfere with protein turnover and protein folding, such as translation inhibitors, proteasome inhibitors, and probably the most striking phenotype was temperature sensitivity. So these phenotypes to us suggest that there's some proteotoxic stress, and obviously the next question is, well, what is the origin of the phenotype? And since there appeared to be stress on the protein quality control pathways, the question we wanted to ask is, well, are these extra chromosomes active? And at the gene expression level, that's in yeast a fairly trivial experiment to do. I apologize that it's a little dark, but I think what I'm showing you here is a gene expression analysis of these disomic strains. And again, we're ordering the genes with position in the genome, along their chromosomes. And if something is red, that means it's upregulated. And I hope you appreciate that the majority of the genes that are present on the additional chromosome are indeed upregulated according to gene copy number. So we can say with a great degree of certainty that indeed these genes are transcribed. The question is, of course, are they translated? And so to address this question, we conducted a SILAC, a quantitative mass spectrometry analysis in collaboration with Steve Gigi's laboratory, especially NOAA 4, to show you actually how impressive this methodology is. I'm first showing you again a CGA analysis of a strain that has 2 copies of chromosome 2. So you see here this whole stretch of the genome is amplified 2-fold, so it's indeed a disome 2. When we look at the RNA level, we start seeing there's some noise. That's biological noise. I told you there's a transcriptional response associated with the aneuploid state, but it's still pretty clear that the majority of the genes present on chromosome 2 are transcribed according to gene copy number. And the same thing is true for the protein. So again, the noise becomes greater. We're still in the process of figuring out how much of this is due to technical problems with the mass spec and which of those are real. But again, generally speaking, the proteins are being made. And we've now conducted this analysis with a large number of different disomes, and it appears that all of the chromosomes are largely active. So I think in sort of an interesting side note that we are interested in exploring is you can obviously now start asking the question, well, which proteins are not upregulated according to gene copy number? We estimate it's about 10% of the yeast genome that is not upregulated according to gene copy number. And as we originally sort of suspected, it turns out that proteins that function in large obligatory protein complexes, like the ribosome, where stoichiometries are especially important, we see strong post-transcriptional regulation at the gene expression levels for these proteins. For example, all ribosomal proteins show what I refer to loosely as dosage compensation. Even though there's 2 copies of the gene, there's actually not 2-fold more of the protein. So we're actually in the process of figuring out which proteins there are. But for the discussion here, what really matters is that I think these data convincingly show that the majority of the genes located on the additional chromosomes are active. And so that would be, and so if these proteins really are what causes these phenotypes, a clear prediction of that hypothesis is is that if I put in large amounts of non-yeast DNA or large chunks of non-coding DNA into these yeast strains, we shouldn't see the same phenotypes. And actually in yeast, we're able to do these kind of experiments. We can use yeast artificial chromosomes. Those are pieces, these are yaks that contain mouse DNA or human DNA. They are the size of a yeast gene, size of a yeast chromosome. They don't make basically hardly any proteins because the splicing and gene expression in yeast is quite different from mammalian cells. So then we asked, well, what happens if we put these yaks into yeast? And the result was actually very clear-cut. I'm not showing you the data. I'm just summarizing them. There was no cell cycle defects, and there were no sensitivities to these proteotoxic stresses, strongly indicating that it's indeed these proteins that are being made from the additional chromosomes that are causing of the phenotypes. So we concluded that the phenotypes that we observe in the aneuploid strains are due to the proteins that are being made, or the transcripts being made from these additional chromosomes. And sort of that led to the hypothesis, and that's still something we're working on, but it led to the hypothesis that aneuploidy results in imbalances in protein stoichiometries. And we suspect that this causes an increased burden on the protein quality control pathways, an increased need for energy to actually get rid of them. It takes actually 400 ATPs to degrade one protein through the proteasome. It's actually getting rid of proteins is a very costly process. And we suspect or speculate, with the evidence for that is meager, I should say, that perhaps some of the other phenotypes that we see that are shared by the aneuploid cells could also be part of this, what we call an aneuploidy stress response. So let me illustrate what we mean by that in this cartoon form here. Imagine there's a protein complex, two proteins that function an obligatory protein complex. They are encoded by two genes located on different chromosomes. And it's actually quite tricky for a cell to make exactly 10 subunits of protein A and 10 subunits of protein P. That's actually quite hard. So the way the cell solves this problem is they make approximately 10 of each, okay? And then use protein quality control pathways such as chaperone-mediated protein folding or proteasomal degradation to sort of neutralize the unassembled excess proteins. And of course, sometimes there's all feedback mechanisms have been described to help establish these stoichiometries. And there's a lot of very wonderful examples for that in the literature: alpha and beta tubulin, ribosomal subunits, histones, and so forth. So what happens, we speculate in an aneuploid cell, is that you're greatly exaggerating this problem, right, of having excess unassembled subunits. So instead of having just a little extra, there's a lot more extra for a lot of different proteins. So we speculate that this causes an increased burden on the protein quality control pathways of the cell. Obviously, increased need of energy. You actually have to make these proteins and then also get rid of them. And we speculate, as I said, that's a speculation, that perhaps the cell cycle delays and other phenotypes are also a consequence of that proteotoxic stress. But this last part needs working out. So we thought that this was true in yeast, and when I told my yeast friends about this idea and this fact that aneuploidy is really bad news for the yeast cells, they went, well, duh. Sort of, wasn't they kind of expected that. And when I told my cancer friends in the cancer center about them, they said, oh, that's really interesting, but I'm sure that's not true in other cells. It must be a yeast thing, okay? So at one point, we decided, and I should say probably getting Howard Hughes money also helped in that decision. We decided we should put our money where our mouth is, meaning yeast people always say, well, if it's true in yeast, it has to be true in the mouse also. But I think at some point, you got to sort of go there and actually test it. And so we decided to bite the bullet and decided to sort of also look at this problem in the mouse. And to generate these defined aneuploidies in the mouse, we took advantage of these very old Robertsonian translocation chromosomes, by which, again, through some Baroque genetics, you can actually generate in a non-Mendelian fashion these trisomic embryos. So in the mouse, trisomy is lethal. All trisomies are lethal. And so we isolate these various embryos at different shortly before they die, and we've done trisomy 1, 13, 16, and 19. We then do, do spectral karyotype analysis to indeed confirm that these embryos are trisomic, and that's an example. These are these Robertsonian fusion chromosomes. So this is, there's 2 copies of 6, there's 2 copies of 17. I hope you appreciate there's 3 copies of 16. So we do SCI to make sure that they really are indeed trisomic. And then the first question we wanted to know is, well, are these chromosomes, like in yeast, are they active? And the answer is yes. What I'm showing you here, at least at the RNA level, they are active. We have not been able to do the proteomic analysis on these cells yet. What I'm showing you here is a gene expression analysis. And what we've done here is we are binning the transcripts, again, by location on the chromosome. So these are all the transcripts on chromosome 1, 2, 3, 4, and so forth. And I hope you do appreciate that the transcripts that are present on chromosome 1 actually are upregulated. So again, generally speaking, not every single transcript, but generally speaking, these chromosomes are active, and genes are expressed according to gene copy number. And so we then, of course, repeated our phenotypic analysis as we have done in yeast. And the only thing I want you to take away from this slide is that the phenotypes we observe in these trisomic mouse cells are remarkably similar to the phenotypes that we see in yeast. So proliferation is impaired. These cells grow very poorly in culture. There's signs of increased need for energy. Glutamine uptake, which is the major carbon source for the TCA cycle in tissue culture cells, is increased in these cells. There's also a sign of, and there's some other metabolic changes in these cells. There's a sign of proteotoxic stress. For example, the protein chaperone, HSP7, 70 expression is increased in these cells. There's also increased levels of we also observe increased levels of autophagy in these cells. So basically, from these experiments, we formulated the following hypothesis. We believe that there's an aneuploidy stress response that exists both in yeast and in the mouse, and we suggest that it's at least in part caused by protein stoichiometry imbalances. These imbalances elicit a response that's aimed at correcting the situation. And again, I should say, I want to emphasize this again, the point that I made at the beginning of my lecture, that this really does not apply to the condition of polyploidy, where obviously stoichiometries are not affected. Okay? So we're clearly still very much interested in characterizing and analyzing these phenotypes caused by aneuploidy in much more detail. But as I also told you in the beginning, we're very much interested in identifying pathways and mechanisms that perhaps help deal cells with the adverse effects of aneuploidy. So I want to switch gear and sort of tell you about that strategy a little bit. I'll tell you about a strategy to isolate mutations that allow cells to tolerate aneuploidy. And we've done this analysis so far in yeast. And basically, we use a very simple strategy. We take a flask of poor-growing disomic yeast strains. We grow them until they grow fast. Then we single colony purify these isolates, these evolved isolates, and then subject them to whole genome sequencing, and then basically look at the various karyotypic changes, and then try and identify driver mutations. So this is sort of this evolution experiment. In red, you see the doubling times of these various aneuploid yeast strains. And then in blue, you see the doubling times of these evolved descendants. And in many cases, we actually discovered dramatic changes in doubling time. And we basically characterized them further. So obviously, what we see a lot is that some of the strains lose parts or all of the additional chromosome. That's perhaps not very surprising. What we see a lot is sort of homologous recombination between repetitive elements in these strains that are scattered throughout the genome. They throw out large chunks of the additional chromosome, and that obviously makes them feel a lot better. But actually, surprisingly, a large number of them maintained their karyotype by CGH analysis, and instead what we found is somewhere between 2 and 8 point mutations in these strains. And so for all the graduate students that are here in the audience and who believe that the omics approach to the world will change everything and will make everything easier, think again. So of course nowadays, identifying, all the mutations in this evolved yeast strains takes, 3 days. You make the DNA, you send it for sequencing, you get this DNA back. So first of all, when you first get that DNA back, to put it in perspective, first, you come back and you filter out all the parents and whatnot. You come back with 5,000 SNPs that you then basically, in the end of the day, have to go through manually and call whether you think they're real or not. You end up with 20-something mutations. Of course, they're all in essential genes, so you can't just knock it out and see whether you can repopulate the phenotype. Instead, you have to recreate that point mutation. And if you're unlucky, one single point mutation doesn't do anything to you, and maybe you need a combination of 22 to sort of recreate your phenotype. So the discovery aspect of the biology is greatly facilitated. I completely agree. But the work, by no means, as far as I can tell, the work is even more. Because when I was a graduate student, I did a genetic screen. I ended up characterizing one mutant, and that was my PhD project, and I got 2 very nice papers out of it. Nowadays, you have to sequence 19 evolved strains, and the reviews itself still tell you, it's not interesting enough, okay? So the omics is not the solution to all your problems. We learned this. I mean, I actually didn't have to learn it. I always knew it, but my knowledge was reinforced by doing this. So something interesting did come out because, we were lucky in a sense. We got lots of different mutations. We got 43 SNPs that have turned out to be real and so forth. And then obviously you have these heap of mutations, and then you have to decide which one of them you're going to study. So that, already that is a big challenge. And, it's quite interesting. The hardest thing, I think, for a human being to do is actually to choose. Because there's always this sort of thought in the back, oh, my God, what if I picked the wrong thing? What if the mutant that I'll give to the younger graduate student is actually the really interesting mutant? And I ended up studying the really boring one. So anyway, at least in this case, we were helped in deciding what we should study. Because what was very exciting is that 3 types of genetic alterations occurred independently in different evolution experiments. And obviously, if you get one thing more than once, you start feeling good about it, thinking, this could be, there could be a causal relationship between phenotype and mutation. And what we got is we got the truncation mutations in this deubiquitinating enzyme UBP6 in evolution experiments for disom 5 and for disom 9. We got point mutations in this vacuolar sorting protein VPS64 in strains disomic for chromosome 9 and 11. And we actually found a duplication of the left arm of chromosome 13 in actually that encompasses several hundred genes in actually 3 different evolution experiments. So for purely historical reasons, we started focusing on UBP6. It was the first one we got twice. So we decided to drop everything and sort of start studying this. So I can't tell you anything about these 2 other genetic alterations. Obviously, we're very interested in them, and if somebody wants to come and study them, find me later. So what I'm going to do is I'm going to tell you about our studies on UBP6. So UBP6 is a deubiquitinating enzyme, and wonderful work by Dan Finlay's laboratory has shown that this enzyme is associated with the proteasome and that it competes with the proteasome for substrates in both a catalytic and non-catalytic manner. So when we read up about UBP6 and saw what Dan's work suggested, I mean, that obviously, just these facts immediately suggested a hypothesis. In the absence of UBP6, flux through the proteasome is increased, and that sort of, in some way that we don't understand, could then help these disomes to grow better. Okay, so that was the idea. So first question, obviously, we had to ask is, are mutations in UBP6 actually causative? Do they improve the fitness or the growth properties of the strains that we isolated them in? And so to address this, we've done what I'm showing you here is a competition assay. We also see when we simply measure doubling times. So we have a wild-type disom 5 strain that's wild-type for UBP6 that's in green, and a disom 5 strain that's where UBP6 is deleted. They're all marker matched and so forth. We just co-culture them and then sort of measure the contribution of the red cells and the green cells in the culture over time. And we were very excited to see that deleting UBP6 indeed improved the fitness of these disom 5 strains. That was good because we isolated it in these evolved disom 5 cells. And it also improved the fitness of disom 9 cells, so that was also good that we also isolated disom 9. But to our excitement, we also found that in addition to sort of suppressing the proliferative defects of these disom 5 and 9 cells, it also improved the growth of disom 8 cells and also of disom 11 cells. And in the others, it didn't do anything. There was only one disom where it actually decreased the fitness, and that was these cells that were disomic for chromosome 2. So this was exciting. So these results suggested that deletion of UBP6 suppresses the growth defects of 4 different disomic strains. And the question then, of course, is, well, how does it do that? And we've done a lot of experiments that I'm just summarizing here. There is one. I'm just not going to tell you this. So, but the hypothesis, as I said in the beginning, that the studies by Day and Finley on UBP6 immediately suggested was that perhaps deletion of UBP6 affects the protein composition of disomic cells, maybe making it more like that of wild type again. And remember, we've been able to quantify the proteome of yeast cells using style-like approaches, so that's exactly what we did. We looked at the proteome of disom 5 cells and compared it to disom 5 cells that lack UBP6. And so what we've done here is I'm showing you the distribution of a wild type versus a wild type. Ideally, that should be 1, that ratio. And but of course, you get some Gaussian distribution with a certain standard deviation. And the first thing you do when you notice when you do that with disom 5 over wild type, obviously, there's all the disom 5 proteins, but that's where chromosome 5 is, and sort of the standard deviation increases, indicating, things are happening in these disom 5 cells. What was interesting is sort of see this, just looking at the data from 10,000 feet above, when we now look at the distribution, the standard deviations of disom 5 UBP6 cells, you notice that that standard deviation decreases, okay? Whatever that means, right? Something happens, okay? So the question is, what happens? So to do this, we decided to sort of dig in the data a little deeper and sort of generated bins for the data. So we basically created 3 bins, proteins that don't change very much in disom 5 cells versus wild-type cells. So that's the that are basically within 1 standard deviation of the distribution. But that's obviously the majority of the cells, right? There's nothing much happens to them. Then there's a smaller group of proteins that are upregulated, they're increased in the disom 5 strains relative to wild-type. That's like 153. And then there's a group of proteins, a little higher, somewhere around 300 genes, whose protein levels are downregulated in disom 5 strains relative to wild type. So we created these 3 bins of no change, downregulated, and upregulated, and then asked, when we delete UBP6, what will happen to these 3 bins, okay? And there the results were actually quite striking. So the majority of the proteins that don't change in disom 5 cells, when we delete UBP6, nothing happens to them, nothing changes. But when you I'd like to draw your attention to the proteins that are upregulated. So excuse me, we binned them and then we calculated the mean. I'm sorry. So this is the mean of these bins, okay? And so what I hope you appreciate is that when we look at the mean of the bin of the proteins that are upregulated, when we now delete UBP6, you get attenuation, okay? And UBP6 on its own doesn't do anything. Interestingly also, the same attenuation appears to occur in the proteins that are downregulated. Okay, so very clearly what deletion of UBP6 does here is it attenuates these effects of the disomy 5 on protein levels. We did in parallel a gene expression analysis to ask, is this attenuation due to transcriptional level or post-transcriptional levels? And the answer was quite clear. This attenuation that occurred for the proteins that were downregulated and were now upregulated, we saw that at the transcriptional level too. So most likely that's some indirect effect, some indirect transcriptional effect deleting UBP6. What we saw at the protein category that were upregulated, so in this case is mostly it was actually due to post-transcriptional effect, indicating that what happens with this class of proteins is they are probably getting eliminated by some post-transcriptional mechanism. We suspect it's by protein degradation, although we cannot exclude the possibility that it's by translation. Okay, so, but I suspect that at this point this is really a formal possibility. So what does this mean? So basically what we think is going on is that, and that's in coordinates with Dan Finley's data, is that in the absence of UBP6, okay, in the absence of UBP6, more proteins are getting cleared through the proteasome, okay? And proteins that have stoichiometry problems and all sorts of proteins that get degraded more. And what we think is going on is that in the case of disome 5 and all the other disomic strains that are suppressed by deleting UBP6, proteins that specifically make these disome 6 are among these UBP6 clients, okay? So in this group of attenuated proteins are the proteins that make disome 5 cell 6. And so that's obviously a hypothesis, and obviously what we're now very interested in figuring out is what exactly these proteins are. I should say a key prediction of this sort of client hypothesis is that we should see the same attenuation in disomic cells that are not rescued by UBP6, okay? And the answer to that question is yes. So UBP6 attenuates the protein levels as far as we know, in both disomic cells that are rescued by UBP6 delete and that are not rescued by UBP6 delete, right? Now we speculate that the clients that, of course, are completely non-overlapping in the different disomic strains, in some cases they matter for fitness, and in other cases they do not. Sort of to simplify this, and sort of the take-home message is that our data indicate that deletion of UBP6 leads the proteome of disome 5 cells be more like that of wild type, and we suspect what's going on is in accordance with UBP6 known and described function, that what the deletion of UBP6 does is it increases, in the absence of UBP6, flux through the proteasome is increased, and that helps attenuate the protein composition in the disomic cells. Of course, there's indirect effects, transcriptional indirect effects also that are occurring. Our data suggests that. So it's not as simple. So clearly, we're still very interested in figuring out exactly how UBP6 affects the fitness of these disomic strains. We're still very much interested in figuring out how the other mutations contribute. I should say, all of these strains that we isolated contained multiple different mutations, and we know in the case of UBP6 that only partially restores the proliferative profile of these originally evolved strains. So there's a lot of synergy between the different mutations. We're trying to figure this out too. As I said, this is early days. And so I want to sort of move on now. Do I have time? Yes? Do I have time? Yeah? Okay. Sort of the last part of my talk that sort of addresses the flip side of this question, sort of, can we identify genetic alterations, or in this particular case, I'm going to tell you about compounds that basically exhibit synthetic lethality with the aneuploid state. Okay, and as we're doing that synthetic lethal screens, obviously, in yeast also, but they are not we haven't reached a state where one can safely speak about them in public. Not so much that I'm worried that we're getting scooped. Nobody would ever in their right mind do these very complicated experiments, but I think there's a good chance that still a lot of them might be wrong. Anyway, so we are more confident. I'm confident enough to talk about the compounds. So we've done two types of approaches to identify compounds that selectively kill aneuploid cells. We've done an unbiased screen in collaboration with the ICCP at Harvard, and that screen identified some interesting compounds that I can tell you nothing about. And in parallel to that, we've done a sort of a more hypothesis-driven screen where we took our lessons learned from the analysis of aneuploid yeast strains and mouse strains and asked, where we learned that these cells appear to be under energy stress, proteotoxic stress, and have cell proliferation defects. And so we decided to ask, if we interfere with cell proliferation, energy metabolism, or protein folding or protein metabolism, can we exaggerate the adverse effects of aneuploidy? Can we create synthetic lethality? So we used a number of proliferation inhibitors, proteins affecting protein homeostasis, and proteins and compounds that create metabolic stress. And what was exciting is sort of of these 20 different compounds that we identified, 3 of them appear to be very selective in interfering with the proliferation of aneuploid strains. One of them was the HSP90 inhibitor 17-AAG, and to a lesser extent galvanomycin. We found the autophagy inhibitor chloroquine to show selectivity, and the AMP kinase agonist ICAR, and to a lesser extent metformin, to show these differences. We spent some time characterizing the effects of ICAR on the aneuploid cells, and that's what I'm going to focus this last part of my talk for you. So what I'm showing you here is sort of the effects this ICAR treatment has on these various trisomic cells. I'm showing you here the effects for trisomy 1 and 13. Similar, though less severe effects are observed when we look at trisomy 16 or trisomy 19. These are the smaller chromosomes. They're not, the effects on fitness of these smaller chromosomal trisomies are less severe. But so, for example, what you see here is that in the presence of the highest concentration, or well, let's take a low concentration, of 0.2 millimolar ICAR, wild-type cells actually still proliferate. Okay, the proliferation is antagonized to some extent, but they still grow. The same concentration of ICAR has a dramatic effect on trisomy 1 cells. Not only does it actually inhibit proliferation altogether, these cells actually die in the presence of the compound. And as I said, similar things are seen in the trisomies, and in the other trisomies. And what I want to show you here is sort of the quantification of that result. And what we're doing here is because these trisomic cells already proliferate poorly, even in the absence of compound. What I'm showing you here is we're normalizing all the effects of the compound with respect to proliferative abilities in the absence of compound. So basically, we're already eliminating the proliferation defect that the trisomy itself causes. If we were to include that, these effects actually would be quite dramatic. But so even by taking out the proliferation defect of the trisomy, you still clearly see that ICAR has a much more selective effect on the trisomy than on wild-type cells. And these are the effects that we see with the HSP90 inhibitor. Again, the larger trisomies, trisomy 1, very severe effect. These effects are not as severe with the less sicker trisomies, trisomy 13, 16, and 19. And sort of a very interesting side note here is we appear to see synergy between these compounds, indicating that they hit parallel pathways in these cells. So, for example, if we combine ICAR with 17-AAG, you actually see, this is basically all these cells are treated with 0.2 millimolar ICAR, and then we start trading in 17-AAG. The effects are actually very, very dramatic. We very effectively can kill these aneuploid trisomic cells. So what does ICAR do to the cell? ICAR is basically interferes with ATP synthesis. ADP and AMP start accumulating in the cell, and that obviously leads to the activation of a protein kinase known as AMPK. And what AMPK does is basically the major nutrient and stress sensor in the cell. And what AMPK does is it downregulates anabolic processes and upregulates catabolic processes. So it downregulates cell cycle and protein synthesis and fatty acid synthesis, and upregulates catabolic processes such as autophagy. So the first question we wanted to address is these effects of ICAR that we see, are they mediated by AMPK? So we've done a knockdown experiment, and that's the knockdown we get. Very depressing. I don't know, there's still a lot of protein there. I'm surprised that we saw something. Anyway, so there's some knockdown. You do reduce the activity of AMPK, albeit poorly. But the effects of ICAR on these trisomic cells were nevertheless eliminated, indicating that indeed ICAR appears to function through AMPK in these cells. I should say it looks better on my screen, but yeah, anyway. And so then the question is, well, what does ICAR do? And obviously, one of the things that ICAR does is it induces p53-dependent apoptosis. And we saw that these cells actually died in the presence of ICAR treatment. So we started looking at apoptosis. And it indeed appears to be the case that in the presence of ICAR, these cells are inducing apoptosis. The early apoptotic cells are shown here in this right bottom quadrangle. And I hope you appreciate that these are trisomy 1 cells here, that the number of early apoptotic cells greatly increases in the presence of ICAR, and to a significantly lesser extent in the wild-type cells. So how does AMPK activate apoptosis? ICAR, activates apoptosis. We know that one of the targets of AMPK is actually p53. So we wanted to know whether the apoptosis or the cell death that we observed was p53 dependent. And again, we've done a knockdown experiment in these trisomic MEFs, and that looked better. So we did knock down p53. And again, we largely lost the effects of ICAR on viability of these cells, indicating that indeed ICAR induces p53-mediated apoptosis. So that sort of leads to this very simple hypothesis that ICAR activates AMPK that induces p53-dependent apoptosis in the cells. Simple hypothesis, but it's wrong. And the reason why it's wrong is because I told you in the beginning, we've actually used a number of compounds and threw at these cells, especially DNA replication inhibitors and components and drugs that activate the DNA damage checkpoint. And we know the DNA damage checkpoint functions through p53. And under those conditions, we do not see a selectivity for the trisomic cells, right? There was no differential effects of DNA damaging agents on wild type and trisomic cells. So it can't be as simple as just sort of activating this pathway that induces p53. Something else synergistic must be going on. And sort of to explore this possibility, we asked, well, does ICAR, in addition to activating p53 in these cells, sort of exaggerate some of the stresses that these trisomic cells already experience? And the answer is yes. I told you in the beginning that HSP70 levels are already increased in these trisomic cells. So this is shown here in the absence. There's already increased levels. And when we now treat these cells with ICAR, this is very dark. That's sad. So you actually start seeing that there's higher levels of HSP70 accumulating in these cells. And the same appears to be true with autophagy. So when we look at autophagy in these cells, it also appears that ICAR appears to exaggerate elevated levels of autophagy also. So we're taking cells here, we're treating them either with or without autophagy, and then we're looking at one of the markers, which is the formation of these GFP-LC3 foci in the cells, which is one indicator of autophagy. We looked at other levels too, and I hope, again, you can appreciate the autophagy is greatly stimulated. So this is obviously all very early on, but our current working hypothesis is as follows. what we believe is going on is that what ICAR does in these trisomic cells, it creates energy stress that leads to activation of AMPK. That in part leads to induction of p53 that contributes to apoptosis. And then in addition, this energy stress that ICAR creates basically leads to the exaggeration of these other stresses that these aneuploid cells experience, proteotoxic stress and metabolic stress. And our big speculation at the moment, okay, and that's a speculation, that's clearly something we're very interested in following up, is that these types of stresses also contribute to apoptosis. And it's the synergy between AMPK-mediated apoptosis and this other apoptosis that together contribute to the selectivity of these cells. Obviously, that's a working hypothesis. There's a lot of dots and question marks in this line. So this is where we're at right now. I sort of gave you an overview of what we're doing. And sort of to summarize, I think our data very clearly indicate that aneuploidy causes a proliferation disadvantage. And again, this is not true probably for polyploids. I think our data also clearly suggests that during transformation, the disadvantages that are associated with aneuploidy need to be overcome in order for a cell to benefit from the no doubt advantageous effects of aneuploidy. For example, it's quite clear in evolution experiments, antifungal resistance, that under those conditions, aneuploidy, disomy of a particular region of the chromosome leads to fluconazole resistance. So, for example, aneuploidy could be an incredibly important in colonizing niches or adapting to new environments. Of course, it can give you an extra copy of MYC that causes incredible proliferative advantages. What we have to bear in mind is at the same time, this aneuploidy comes with baggage, right? And we have to evolve strategies to sort of deal with this, with the bad effects in order to, take advantage of the benefits of aneuploidy. I think our data also raised a very distinct possibility that cancer cells may more heavily depend on the mechanisms that help cells deal with the stresses that are associated with aneuploidy for survival. I think in the light of this idea, it's very interesting to note that proteasome inhibitors are quite successful in the clinic. And way back when we were all cell cycle researchers, we were actually very surprised that it did so well, right? Just inhibiting the cell cycle, why should that make such a big difference? I think our studies on aneuploidy might provide a framework to think about these Velcade effects. And I think one possibility that we are very excited about and hope to explore further is that this idea that the phenotypes that are shared by the aneuploid cells actually may provide new opportunities for therapeutic targets. And our studies on ICAR and HSP9 inhibitors could sort of provide one way of going forward. So in the last minute of my talk, I want to sort of come back to this question of the role of aneuploidy and its relationship to cancer. So I spent now the last hour trying to convince you that aneuploidy is bad news for the cells. And I think you should all now say, well, wait a minute. If aneuploidy really antagonizes cell division and is so bad for the cells, why then are most tumors aneuploid? Okay? And as I said, there's definitely benefits to it. And I think you can look at, there's sort of two ideas that are out there. And I like both of them. I like the second one better. So the first one is sort of best characterized by a German proverb that sort of goes, among the blind, the one-eyed is king. And basically what it means is everybody around you cannot see, okay? And you're the only one who has, albeit only one eye. Nevertheless, you're top dog, okay? And sort of the idea is that in a tissue of completely terminally differentiated cells, okay, if you through a non-disjunction event got an extra copy of chromosome 13 with MYC on it, okay, even though you will proliferate really poorly because you have all this extra baggage of this one extra chromosome, MYC will give you an edge, okay? An extra copy of MYC can give you an edge. And so you can, in this sea of non-dividing cells, start dividing. So I think that's a definite possibility. I think it's definitely true for terminally differentiated tissues like the neurons or brains and so forth. I think that argument becomes a little more tricky if you have tissues with high turnover rates, such as the colon, the skin, and so forth, right, where these cells get shed very quickly. And so so we are entertaining sort of this second idea, and we actually have some evidence in yeast to suggest that that might be true, that aneuploidy causes protein imbalances, that causes stress and affects, DNA replication, repair, chromosome segregation pathways, and then actually leads to genomic instability. And this is not a completely crazy idea, because I think actually from work in bacteria and actually more recently also in tissue culture cells, there's actually very nice evidence to suggest that in stages of stress, mismatch repair machinery gets downregulated, translesion polymerases get upregulated, and cause increased mutation rates and all sorts of forms of genomic instability. And what we actually see in almost all of our disomic yeast strains is they all have defects in homologous recombination. And obviously we're now quite interested in figuring out where these repair problems come from, and they also show increased mutation rates and increased chromosome loss rates. So that is perhaps one way, rather than thinking of the aneuploid state as an oncogenic event or a tumor suppressing event, you can actually think of the aneuploid state as a mutative phenotype. Sort of it allows you to roll the dice more frequently in order to sort of evolve out an incredibly fit new cell that either proliferates very well or that's able to colonize distant sites or new niches. So with that, I want to acknowledge all the people who've done the work in this lab. The aneuploidy work in yeast has been started by a very talented postdoctoral fellow in the lab who's going to be on a job market, Eduardo Torres, who's been added by a graduate student in the lab, Christian Gonzalez. All the yeast work is a long-standing collaboration with Maitreya Dunham at the University of Washington, Seattle. All the computational and gene expression analysis was and continues to be a wonderful collaboration with Maitreya. And then the mouse work, pioneered by a very brave postdoctoral fellow in the lab who started to work on mice in an exclusive yeast lab. So he gets all the credit, also on the job market, if you're interested. Vineet Prabhu, he was a brave graduate student who joined Brett. And all the work on ICAR that I've been telling you in the last part of my talk has also been done by a very talented postdoctoral fellow in the lab, Yun-Chi Ting. And with that, I thank you for your attention. You want to call?
Yes. You said all your aneuploid yeast strains have defects in homologous recombination?
Not all of them, a lot of them.
Okay. What kind of defect?
I have no idea.
Are they hyperrec or hyporec?
They don't have they have a repair defect. So RAD52 phosphate. That's all we know at the moment.
Yes.
With respect to the real tumors, do you ever pick up as suppressors of the slow growth of the aneuploids? Do you ever get spontaneous tetraploids?
Never.
No. That's not a way that they speed up.
So I have some thoughts about tetraploidy and where this sort of near tetraploidy comes from. So first of all, I want to be very clear. There's no such thing as a tetraploid tumor. It is true that many tumors have a near tetraploid chromosome content. But if you do sky analysis of these chromosomes, they're all over the map. They're monosomic for chromosome 1, 5 copies of chromosome 7, and they are not tetraploid. I think what this near tetraploidy comes from is a limitation of the mitotic spindle. I think there's only so many chromosomes that you can actually get onto a spindle. So that in the end of the day, it sort of ends up that that's how many chromosomes you end up with. But there's no such thing as a tetraploid tumor. I think that's actually a terrible misconception in the literature. And I think by now people should know better, right?
The thought was just that if you could balance everything out better.
So you're absolutely right. You're absolutely right. I should take this back. You're absolutely right. No, I should, no, not that part, actually. I was thinking about something that I said before. If we have a haploid plus 1, that's significantly less fit than a diploid plus 1, which is exactly the prediction of the protein stoichiometry issues. So you would think that, if you make it 4n plus 1 or 2n plus 1, that should make it better. In our yeast evolution experiments, with the exception of one disome E4, we never see that happening. I think it's a good question. Maybe
If you did the colchicine trick and you made them to
yes, so that, but we already know that they are better because 1n plus 1 is more sick than 2n plus 1. So, but I think it's an interesting question of why wouldn't you get this in evolution experiments? And I think what could be going on there is that there's sort of an unusuality of the way cytokinesis works in these. So in mammalian cells, if you have problems during mitosis, you get ingression and then you get deregression, you become tetraploid. So quite easy to do, right? In yeast, you make your site of cytokinesis in G1, and then you have to sort of wiggle the nucleus through. So, you cannot sort of get rid of cytokinesis. You can't reverse cytokinesis just by the way yeast cells are. So I think it'd be a very interesting question to ask in the MEFs. Yes. Yes?
You know what I'm going to ask. So have you tested for what mitochondrial function, how that feeds in either of your systems?
It's a wonderful question. And we should do these experiments better, okay? The only thing, obviously, respiration, energy, what's the respiratory state of the cell? How are mitochondria doing? For example, F1 ATPase and all these large protein complexes, you would think that, stoichiometry problems in these could affect respiration, that could cause stresses in energy. It's a great question, Jody. The only thing I can tell you is our yeast cells can grow on glycerol as a sole carbon source. That doesn't obviously tell you whether there's some compromise in respiratory.
Converse to that, how about rosera cells?
So we haven't. Yes, it's a great question, but we have not addressed it yet. Right? Yes.
I'm just wondering, in tumor cells, can you speculate how aneuploidy arise from normal cells? Do you think they go through a stage like a tetraploidy? If they do, does aneuploidy have growth advantage compared to tetraploidy or perfect ploidy cells?
So I think it depends on the tumor. I think that's quite clear. I think in the colon and in the breast, where the and I think in melanomas too, sort of what is considered to be pre-malignant lesions, okay? They have low-grade aneuploidies, okay? There's like 1 extra chromosome, 2 extra chromosomes. It is nothing as severe as you see in the later stages, okay? Cervical cancer is quite different. There, you get very, very high-grade aneuploidies very early on. So now you're going to ask me, what's the difference? The difference, I believe, is the p53 status. Because in cervical cancer, when the virus HP, the first thing it takes out is takes out p53. And there's some very nice work by David Pellman by Bob Mangalis that suggested that there is what's called a tetraploidy checkpoint that's p53 dependent. Well, when cells go tetraploid, they die, and in a p53 dependent manner. Okay, so if you want to go through the tetraploid intermediate, you kind of have to be p53 minus. So in tumor viruses like HPV, it's very likely that that is the path to tumor, to aneuploidy. However, in most epithelial human cancers, loss of p53 is usually a very late event. And it's usually then when you start seeing these crazy karyotypes. So I think these highly abnormal karyotypes are really a direct consequence. That's my gut feeling, is a direct consequence of loss of p53. Yes.
It's been shown that the aneuploidy is fairly common in yeast and biofuel yeast strain, wine and biofuel yeast strains. Have you guys looked to see if those?
No, I think that would be a
those mutations are-
it will be a tremendously exciting thing to do. And actually also in plants, you know. Although in plants, the reason is probably because there's a lot of post-transcriptional gene silencing going on and buffering. But in these yeast strains, that's probably not going on. So I think that'd be something very exciting to look at.
What was the nature of the mutation in that gene as well?
UEP6, you mean? It's an interesting mutation. It's a premature stop codon that truncates the peptidase domain but leaves the ubiquitin ligase domain intact. The ubiquitin binding domain intact. But in all our analysis, we looked at the truncations, the catalytic dead, and the deletions. They all behave the same. So we believe that it's loss of UBP6 function that creates that phenotype. Yes?
So there are a lot of cancer genomes now sequenced. Do you see then the same themes that you would expect from the analysis in yeast as to what mutations you would select for to give them an aneuploidy growth advantage?
So we've identified 43 mutations, in our different yeast strains. Mind you, we've only shown for one of them causality. All the other ones, they could all be passages. However, it's interesting to note that some of those have been found mutated in some cancers. Do I know what that means? No, I don't. But, there is potential. That's all I can say is there at this time. Joanne.
So what's the phenotype of UBC6, just in wild
On
type
its own,
cells?
there's no phenotype at all.
So the wild type cells has no problem with that.
Nothing. No. So under conditions when you start, when ubiquitin becomes limiting, then obviously these cells will start, because they have actually the major phenotype that these cells have is that they lose ubiquitin very quickly, because they actually don't take the ubiquitin chains off the substrates. They get sucked into the proteasome and degraded. And so under conditions where ubiquitin is limiting, these cells start having phenotypes that are indicative of loss of ubiquitin function. But in normal wild-type cells, no phenotype, no.
And then I want to ask a question about the inhibitor. So is your prediction then, and I'm a little confused on your
Inhibitor?
The inhibitor in the
ICAR?
mouse.
That's actually an agonist. Activate.
Okay,
Activates. It
okay.
activates ADA.
Would your prediction be that that wouldn't function in P53 in tumors that lost P53?
Yes, so that's obviously
Why
depressing.
do you-
Thank you for pointing this out. So that's obviously the depressing part about this discovery, right? Because obviously most tumors are P53 minus. So if ICAR only kills aneuploid cells that are P53 plus, well then obviously the potential therapeutic value of that drug is 0.02 or something. I mean, whatever the percentage of P53. 40% of tumors are P53 positive?
60 or P53.
Oh, that makes me feel a lot better. Okay. Oh, okay. Excellent. Well, there you go.
So on that pathway, you suggest that there
So that's wild speculation. Okay, I have no evidence for that except that it was a need to explain of why DNA-damaging agents that also induce P53 don't show the selectivity whereas ICAR
But
did.
when you get rid of P53, the effect all goes away, correct?
Yes. So the genetic argument would be that you need both, right, in order to see selectivity. But that actually does make me feel a lot better. Okay. Thank you very. Well, it's getting late, right? I'll talk to you later. Thank you very much.